Hello, good morning. Welcome back to Daily Lectures by New Age Cosmetic White EdTech Healthcare Consultancy. I am Dr. Bobby. We're on the subject of molecular biotechnology and the chapter on large-scale production of proteins from recombinant microorganisms. So now we've reached the summary. The summary states that the large-scale production of genetically engineered organisms in industrial-sized bioreactors greater than 1,000 liters is not achieved merely by extrapolating directly from laboratory growth conditions, zero point 0.1 to 1 liter, the temperature, pH rate, and nature of mixing oxygen demand for aerobic organisms and nutrient levels must be taken into consideration when large bioreactors are designed. The temperature, pH rate, and nature of mixing, oxygen demand for aerobic organisms, and nutrient levels must be taken into consideration when large bioreactors are designed. Microbial fermentations can be performed in several different ways. In batch fermentation, an inoculum of cells is added to fresh medium and the fermentation is allowed to proceed without supplementation until the maximum amount of the target product is synthesized. Under these conditions, the cell culture passes through 6 phases of growth: lag, acceleration, log, deceleration, stationary, and death. Protein production is optimal during the log phase, whereas peak production of many low molecular weight products occurs during the stationary phase. It is important to monitor batch fermentations closely to ensure that the cells are harvested at the appropriate time. In fed batch fermentation, growth medium is added at levels or various levels, usually to prolong the log phase of the fermentation process. Continuous fermentation entails adding fresh growth medium throughout the course of the fermentation and simultaneously removing cells and spent medium. Each of these fermentation systems has disadvantages and advantages for large-scale production of recombinant products. Although continuous fermentation is still a relatively untried industrial process, the approach has some inherent benefits, making it likely that its use will become more widespread in the future. One way to increase the amount of recombinant protein is to grow transformants to as high cell density as possible. The best way to obtain high-density cell cultures, is to use a fed batch fermentation strategy. Fed batch fermentation has been directly compared with batch fermentation for the production of several different proteins, and in all cases examined, fed batch fermentation has resulted in a higher rate of target protein. To obtain the largest amount of product in a given volume, it is helpful to attain high cell densities to avoid the loss of recombinant plasmid, to utilize E. coli cells that can become poisoned during foreign gene expression, to secrete the foreign protein into the growth medium, and to avoid the formation of acetate in the medium. There are 3 basic bioreactor configurations: STRs, bubble columns, and airlift reactors. Currently, STRs are used most frequently in industry, but interest in airlift bioreactors is increasing. In STRs, mixing is achieved by mechanical agitation. In airlift reactors, both aeration and mixing are performed by a gas, usually air, that is introduced through a sparger at the bottom of the vessel. And with either the internal baffles or external loops causing the fluid to circulate within the vessel. Bubble columns are similar to airlift reactors but lack the design features that cause the culture medium to circulate in the vessel. The problems of the maintaining sterility, pH, temperature, and other fermentation parameters are overcome in two different ways depending on the design of the bioreactor. Two-stage fermentation processes with either tandem airlift reactors or single STR have been successfully used for the production of recombinant proteins. When the product is present within the cells can be harvested by either centrifugation or cross-flow filtration and lysed chemically, enzymatically, or mechanically. The preferred forms of mechanical cell lysis include wet milling, high-pressure homogenization, and impingement. The product is then fractionated from the cell lysate. Ultrafiltration has been found to be an effective method for large-scale dialysis, concentration, and initial fractionation of proteins produced by recombinant organisms. Scientists have begun to establish procedures for the large-scale isolation of plasmid DNA. These procedures must take into account the host cell and its growth and metabolism, plasmid size, cell lysis methods, and the complete removal of a number of potentially contaminating cell components. And now onto the review questions. What are the differences between batch, fed batch, and continuous fermentations? How has fed batch fermentation been used to improve the production of insulin B peptide, interferon gamma, and Fab fragments? What parameters must be monitored and controlled in an optimized fermentation process? What is the effect of a recombinant plasmid on the growth of microbial cells? How does the mixing of a growing microbial culture affect the transfer from the growth medium to the cells? What are the advantages of a high cell density during a large-scale fermentation? What conditions lead to a high cell density during a large-scale fermentation? What strategies can be employed to prevent acetate inhibition and the growth of recombinant E. coli strains? What are the relative advantages and disadvantages of using an STR or an airlift fermenter? Compare the growth and induction of a recombinant microbial culture using 1, 2 reactors in tandem and 2, a single reactor. What is downstream processing? What strategy would you use to verify a recombinant protein that is secreted into the growth medium? What are the advantages and disadvantages of large-scale mechanical lysis of cells compared to chemical lysis? How are cells mechanically disrupting using 1, wet milling, 2, high-pressure homogenization, and 3, impingement? How are microbial cells concentrated at the fermentation stage of a biotechnological process? What are the advantages and disadvantages of these procedures? How are the large volumes of protein solutions partially purified using ultrafiltration? How can the activity of an insoluble recombinant protein be recovered? What factors should be considered for the large-scale isolation of plasmid DNA? How can quiescent E. coli cells be engineered to produce large amounts of foreign protein? What strategy would you use to ensure that a plasmid encoding a larger target protein is not lost during the large-scale growth of a recombinant bacterium? What are some of the advantages of secreting a recombinant protein into the growth medium? And now I'm going on over to the start of the subject to give you a glimpse of all the illustrations so that you can just make your research into what you've learned or listened to so far. The first illustration would be figure 17.1, which is a generalized scheme for a large-scale fermentation process. The commercial product is usually in either the cell or cell-free fraction, but are not in both. Consequently, one or the other of these fractions will be processed further or discarded. Excuse me. Have a look at the illustration, take screenshots and do necessary research, right? That's it about that there. The next one. This is figure 17.2, which is a schematic representation of the time course progressive curves of cell concentration, mass, and substrate concentration in batch A, fed batch B, and continuous C fermentations. That is the illustration here, the representation. And this is figure 17.3, which is pattern of microbe growth, cell growth in a batch fermenter. The 6 phases of the growth cycle are 1. Lag, 2. Acceleration, 3. Log, exponential, 4. Deceleration, 5. Stationary, and 6. Death. Take a copy of this and please research onto it. This is a schematic representation of bacterial cell in which the essential protein synthesis initiation factor, one gene in for A, was deleted from the chromosome and included in plasmid. The target gene is under transcription-led control of the strong PRC promoter. PRC is controlled by the lactic repressor encoded by LAC. LQ, which overproduces the protein. Have a look at this. Take screenshots. The next is a synthesis of a single-chain antibody fragment, SCFV, in quiescent E. coli at 30 degrees centigrade. And the CL repressor encoded by CL1857 inserted into the chromosome from DNA binds the operators of the PR and P1 promoters. PL and prevents transcription of plasmid encoded. SCFV and RCD genes. At 42 degrees centigrade, the temperature sensitive repressor is inactivated so that transcription directed by the PR and PL promoters succeeds. Turning on the PR promoter causes the RCD protein to be synthesized, thereby causing the cells to become quiescent. At the same time, turning on this promoter activates transcription of the gene encoding SCFV. When the RCD gene is induced, a mutant of the HNS gene causes the cessation of host cell protein synthesis. The next is a cell growth factor and foreign protein SCFV secreted into the growth medium for E. coli cells with induced quiescence compared to control white type cells. You can take a screenshot. This is figure 17.6, which shows engineering proteins for high level expression following high cell density growth. A target protein gene is placed under the transcription control and strong and inducible, in this case by amyloic acid promoter. The target gene is fused to any one of several different secretion signal sequence. See rare codons are removed from protein, following consequences that are not well expressed by chemically synthesizing the entire gene. CSF granular cell macrophage colony stimulating factor. That is the diagram there. This is a figure that is a schematic representation for glucose metabolism in an E. coli strain that has been transformed with a plasmid carrying the genes for the protein subunits of acid or lactate synthesis ALS. Note that there are the conversion of glucose to biomass is a multi-step process CoA and coenzyme A. Now this is the replenishment of the TCA cycle in E. coli by the introduction of a gene from R-ethylene encoding pyruvate carboxylase. This avoids the conversion of pyruvate to acetate. TCA cycle may also be replenished by the introduction of a gene encoding aspartate, converting aspartate in the medium to fumarate. Note that the conversion of glucose to biomass is a multi-step process CoA and coenzyme A. Regarding now, so leave it. And look all over. This is the replenishment of the TCA cycle in E. coli by the introduction of a gene from R-ethylene encoding pyruvate carboxylase. This avoids the conversion of pyruvate to acetate. The TCA cycle may also be replenished by the introduction of a gene encoding aspartate, converting aspartate into the medium to fumarate. Note that the conversion of glucose to biomass is a multi-step process. Table 70.2 shows relative acetate levels, yeast cell yields, and foreign protein activities from E. coli cells transformed with an R-ethylene pyruvate carboxylase gene. This figure shows simplified examples of bioreactor configurations: A. SGR, B. Bubble column, C. Internal loop airlift reactor with a central draft tubes, D is external loop airlift reactor. The arrows within the bioreactors indicate the direction and flow of the culture medium. Next is a two-stage airlift reactor used for the temperature-dependent induction of a protein product. Cells from the growth stage left fermenter, which is at 30 degrees centigrade, are pumped into the induction stage fermenter right, which is at 40 degrees centigrade. The bioreactor has a double external loop that is fitted with valves. By changing the valve settings, what molecules is best suited for different fermentation can be created. This is the diagram for that. Next is a scheme for the large-scale production of the protein A, G, beta, gal. The volumes in parentheses indicate how much culture medium was used at each step of the process. The culture medium occupied about 60 to 75% of the total volume of each of the bioreactor's fermenters. The scheme for large-scale production of the protein A, G, beta, gal. The volumes in parentheses indicate how much culture medium was used at each step of the process. The culture medium occupied about 60 to 75% of the total volume of each of the bioreactor fermenters. This is a schematic representation of the amount of foreign proteins produced as function of time by recombinant E. coli following induction arrows. In the mid-log phases of growth, prior to induction, no foreign body protein is synthesized. After induction batch mode, in the absence of additional nutrient, the cells soon enter stationary phase and synthesize proteases that degrade the foreign protein product. Take details of this, take a screenshot, and it should help you. This is a comparison of batch and fed batch fermentation for the production of fusion protein, including insulin B peptide. This is an figure showing the induced recombinant protein production with batch and fed batch E. coli cells to late logarithmic phase. The cells are either not induced with 0.05 mm IPTG or induced with 2 grams of lactose per liter. Have a look at the diagram, just copy it through with a screenshot. This is a membrane filtration systems for concentrating microbe cells, as you see here. This is a schematic representation of 3 methods of mechanical cell disruption of microbe cells: wet milling, high pressure homogenization, and impingement. This is a schematic representation of large-scale ultrafiltration dialysis of a protein solution. This protein solution from the feed tank is pumped across the ultrafiltration membrane, with only a fraction of the solution actually passing through the membrane and the rest being used to sweep through the membrane clean protein. This is the picture that depicts it. And this is a schematic representation of large-scale protein purification using ultrafiltration membranes, with an example of 2 membranes using frequently sequentially 1000 Ka cutoff membrane and a 10 Kd cutoff membrane in order to purify B 30 Kda protein. Take a screenshot of this diagram, it should help you. This is a schematic representation of a process for the continuous thermal lysis of E. coli cells for the large-scale preparation of purified plasmid DNA. So that's it about schematic representations, figures, and illustrations, along with the review questions and summary. Make good notes, do adequate research, and if you have any doubts or need any clarifications, please come over to the comment section on YouTube or my email ID and here on WhatsApp. Kindly do not commercialize any aspect of these portions. Please use it to the best outcome for your exams and your future careers. Thank you. Stay put. Best wishes. Bye.